Hi Year 12. Let me know if you are here. I can't see any of you in the chat, so please say hey if you are with me today. Hey, great, we've got one person. Hey Aaliyah, how are you doing? Hopefully a few more are going to join. We'll wait a couple of minutes and just see if they do. We're going to do a lesson today on viruses. Now, GCSE, obviously, you do learn about pathogens, you learn about bacteria, viruses, but for A-level, we're also going to learn about the structure of a virus and how viruses can replicate inside a cell. Hey, we've got a few more people here. Hey, Evie. Hey, Delight. Hey, Zafia. Hey, person one. Good morning to you. Hey, right, Lily, welcome. Okay. Hope you've got your workbooks ready to go. You've got your paper, your pen, and you are ready to learn. Let's make a start. Okay, so this is GCSE at this point, right? I just want to know from you, if a GCSE question came up and it asked you to give a definition of a virus, what would you say? Can you name any viral infections that you learn about at GCSE or any others that you know about? Let's just start off with what we already know, right? And then we can build from there. Hi, so Amponza, first time you've been on so far. Have you been just watching the videos? Have you been watching the recordings? Good to have you here live with us today. Yeah, be proud of yourselves. It's quite an early start, isn't it? Right, so what is a virus? Malaria. Malaria is not caused by a virus, okay? Malaria is actually caused by a protist, which is a different type of pathogen carried by mosquitoes, as you probably know, but it's not a viral infection. Okay, but Aaliyah says measles. Yes, measles is caused by a virus. TMV, tobacco mosaic virus. That's one that affects plants, right? It causes, it affects plants. It causes decoloration of the leaves because it reduces the amount of chlorophyll. So yeah, they are two viral infections that you definitely learn about at GCSE. Are there any others? The common cold. That is caused by a virus. Any others that we can think of? What about one that we've talked a lot about recently over the there we go, Mark. Yeah, coronavirus. COVID. Yeah, exactly. COVID is caused by the coronavirus. The flu, which is caused by influenza, which is another virus. Okay, so there's quite a few that we know of. No one has yet given me a definition. Doesn't matter if it's just GCSE, I'm not expecting you to know any more at this point. So what is a virus? Really simply, what would we say? Let me start us off. So it's in the same group as a lot of bacteria, some protists like the malarial protist. What are all these things that cause infection? Okay, Jawahir says a pathogen. That's what I was thinking, is a type of pathogen, which basically means it causes disease. Okay, we should know that from GCSE. So we've got some more comments coming in. So Delight says it's non-living, which is correct. Viruses are not classed as living things. Viruses enter cells. We call them host cells. So you might know that from GCSE. And then Aaliyah says they replicate inside them. Good. That's basically all I would expect you to know from GCSE, is that they get inside like one of our cells, which we call a host cell. They replicate inside that host cell, so more viruses are made, and then they destroy those cells. Okay, because they replicate until there's so many inside the cell that they burst the cell and go back into the bloodstream, and then they can go and infect other cells, so they're destroying cells as they go. For OCRA biology, do you need to know all of the practicals? Yeah, you still have required practicals. Not all of them are the same as the AQA ones, but most of them are. Most of the practicals are similar. There's one or two that are different. But yeah, you still do required practicals, and practical questions still come up on the OCR papers, just like they do on AQA. And you're probably used to that from GCSE, right? You know there's going to be practical questions. Okay, right, shall we move on then? So, a question came up on an A-level paper. I think it was only last year or maybe the year before, and it said to students, viruses are acellular. What does this mean? And it was only a 1-mark question, but a lot of students, more than 50% of students, didn't get a mark because they didn't know what that meant. So what do you guys think acellular actually means? I mean, it's quite simple if you think of the word acellular. What do we think we're saying here? Exactly. Exactly. Oh, we've got some confusing answers now. We've got some opposites. So not made of cells is correct. Okay, they are not made of cells. So Jawahir, they are not cellular organisms. Person 1, they are not just one cell. One cell would be unicellular. One cell is unicellular. Many cells is multicellular, but acellular, let's write this down because there was a bit of confusion there. They are not made of cells, or you could say they are not cells. A virus is not a cell, okay? A bacterium is a cell, but a virus is not a cell. Exactly, no cells. In fact, as you said, they are actually not living, okay? Now, if we go on to explain this a little bit more, why are they not cells, okay? So what we can say is they don't have cellular structures. Okay, hi Maya, you're new as well. So for example, no nucleus. But we can't just say that because bacterial cells don't have a nucleus and they are still considered cells, right? So just having no nucleus is not enough to be classed as acellular and non-living. But as well as no nucleus, they have no cell membrane, no ribosomes, okay? Obviously no mitochondria. So they don't have the same cellular structures, okay? They are not cells. And they also don't do cellular processes. So for example, what is like the main cellular process that releases energy that all living things do? What is that process? Because viruses don't do it. Exactly, Beth. They don't respire. They don't carry out respiration, okay? There are other things as well. They don't grow in size. Yeah, but the main one is like they don't respire. That is, if you think all the way back to year 7, think back to Mrs. Gren. Who remembers Mrs. Gren? Or Mrs. Nerg? Who remembers that? Those letters like movement, reproduction, sensitivity, growth, they don't do these things. They don't grow bigger. They don't respire. They don't need nutrition. So they're not classed as living things, okay? They can become a sort of crystal, right? I don't quite know what you mean by that. They do replicate, but remember guys, they have to be inside a cell to replicate. Yeah, Jawahir, they can multiply, they can replicate, but they have to be inside a host cell to do that. They can't do it on their own, okay? But the big one, I think, they don't respire. So it's kind of obvious they are not living, they are not made of cells, they don't have any of these cellular structures, okay? I think some teachers didn't actually teach what acellular meant because so many students did not get a mark for that question, which was meant to be an easy question. Should we have a look at the structure then of a virus? So viruses all look slightly different, okay? I've given you like the generalized structure of a virus, but they do all look slightly different. The one we're going to focus on is HIV, okay? We're going to focus on HIV today, but let's just label some of the general components of a virus, okay? So two of them are labeled on the diagram for us. So let's do those ones first. So A is what we call an attachment protein. Okay, so there are proteins on the surface of the virus. They're called attachment proteins. Now the yellow bit, it's not a cell membrane, okay, because viruses don't have a cell membrane. It's called, let me call it a viral envelope. Okay, it's made of lipids, but it doesn't have the same structure as a cell membrane, so we can't call it a cell membrane, okay? It's a viral envelope or a lipid envelope is what we call it, okay? Yeah, so A is the attachment protein. If you label the yellow layer on your diagram, that's the viral envelope, okay? Now B is called, does anyone know what B is? You won't have heard this before, I don't think. What is the difference between an attachment protein and an antigen? That's a really good question. They are very similar. They are both proteins on the surface. I suppose with the viruses, we call them attachment proteins because it's the attachment proteins that allow the virus, let's write this down, allow the virus to attach to a host cell, okay? But they are very similar, they are proteins on the surface. I suppose for an antigen, we say it's the protein on the surface of a cell. This is not a cell. So we call them attachment proteins and they're used to attach to the host cell so the virus can infect it, right? Good question. Right, so no one seems to know what B is. So B is called a capsid and it's made out of protein again. Sometimes it's actually called the protein coat. I'll just put that as well, the protein coat, but the scientific term for it is the capsid, okay? Now, that's the little orange beads. They look like orange beads, but they're proteins, okay, the capsid. Now, inside the capsid, you've got these two red squiggly lines. That is the nucleic acid. Can anyone give me an example of a nucleic acid? Well done, Kira. Yes, they are. Can anyone give me an example of a nucleic acid? We don't tend to use this term at GCSE, but you've definitely learned about nucleic acids. Good. So nucleic acid. So it's either going to be DNA, like our cells have DNA, or some viruses, like HIV, which we're going to look at next, some viruses have RNA. Okay, that's why I just said nucleic acid. I was trying to be general because it depends on the virus. Some have DNA as their genetic material and some have RNA. Okay, so this is the genetic material. Okay, and then the pink dots, which Kira has just told us, the pink dots are what we call reverse transcriptase. Kira, how did you know that? Reverse transcriptase, which is an enzyme. How did you know reverse transcriptase? Has anyone else heard of that? Not all viruses have this, okay? Let's make a little note here. So not all viruses contain reverse transcriptase. When I don't do PE, I go to my biology teacher's A-level lessons. That's pretty cool. That's impressive. Be aware though, not all viruses have that, okay? We're going to talk about that in a second. HIV has it, which is what we're going to focus on, but not all viruses do, okay? So you can see very clearly this is not a cell. There's no cell membrane, there's no ribosomes, there's no mitochondria, there's no nucleus. All we've got is a very simple structure. We've got a lipid envelope with attachment proteins. We've got a capsid, which is made of protein. And then we've got some kind of nucleic acid, like DNA or RNA. Okay, so it does have genetic material. What is your question? Put your question into the chat. And I will try and answer it if I can. We'll see. Okay, so this was just kind of a general introduction to protein structure, but on the AQA specification, you only have to know about HIV. What's the purple bit? Nothing really. It's not cytoplasm, it's space. It's literally just a lipid envelope and then a layer of protein inside it. There's probably not quite as much space as this diagram is showing you. What's the capsid again? So the capsid is just made out of protein. And if you're interested in the role, it's basically to protect the genetic material. It's to protect, like the DNA. It's to hold the DNA. It's to hold the RNA. It's kind of protecting that DNA inside. Why don't all viruses have reverse transcriptase? We're going to talk about that now. That's a really good question. So, if you go on to do AQA biology, you are going to have to focus on the structure of HIV. If you're going to do OCR biology, you don't do as much about HIV as AQA. Okay, you still have to know viruses, you still have to know viral structure, but you don't have to know HIV specifically. Okay, so the diagram that I gave you before was basically HIV. Okay, I always use HIV as my example. So we've got, let's just go through it again because I know this can be a little bit confusing. So we've got attachment proteins on the outside. They are attached to the lipid envelope. So we're calling it a lipid envelope and not a cell membrane. Inside there is a capsid. And this is about the amount of detail that you need to know, okay? So we're not going too in depth. But inside there is a capsid made of protein. And inside the capsid, for HIV, the nucleic acid is RNA. Okay, so we're focusing now specifically on HIV. HIV has RNA. Does anyone know what RNA stands for? What's the purpose of the viral envelope? So a couple of things, MZA. Main purpose, obviously it's kind of holding the whole virus together, right? But it's the attachment proteins. The attachment proteins are fixed to the lipid envelope. And the attachment proteins are vital because that's how the virus is going to get into the host cell. Those attachment proteins need to be held in place by the lipid envelope and then they attach to the host cell. Good, we all know what RNA stands for, or Chizzy does. Hassanin does as well. It's ribonucleic acid. Okay. MZA, I've seen them ask for the purpose of the capsid. In fact, I haven't, I'm lying. I've seen them ask students to choose one of the structures and give the function. So you could have chosen the capsid and given the function, but you could have chosen the attachment proteins and given the function. You could have chosen the nucleic acid and given the function. So you didn't have to know the functions of all of the parts. The lipid envelope is the same as the viral envelope. Yeah, let me just put that here. It's the same thing, just two different things that you can call it. Okay, So RNA stands for ribonucleic acid. Now, HIV has reverse transcriptase enzymes, which were the pink dots. Does anyone know, this is a hard question, but does anyone know why HIV would need reverse transcriptase? Does anyone know the role of reverse transcriptase? Because someone asked me before, why don't all viruses need it? HIV needs it, reverse transcriptase, but some viruses don't. Okay, so Kira says to convert the RNA into DNA. You're spot on, to change the DNA. So Jawar here, it's more about changing the RNA into DNA. Okay, so HIV has reverse transcriptase to convert the viral RNA into DNA. That's the job. It does the reverse of transcription. So if you remember what transcription was, which was one of those hard topics on GCSE, but transcription was about making RNA from DNA. So reverse transcriptase makes DNA from RNA, or it converts RNA into DNA. Now, if this cell is gonna, sorry, I just made a big error. I just called it a cell. It's not a cell. If this virus is gonna infect like a human cell, why is it gonna need to convert the RNA into DNA? Think about our genetic material. Why do we need to convert the RNA into DNA? What's inside our cells? Because ultimately, the virus is going to try and integrate its genetic material into our genetic material. Yeah, so Beth says, so it's kind of able to control our cells. Yeah, you're on the right line. Yeah, to make more proteins, which can make more viruses. Yeah, our cells, our host cells that are going to host this virus, they contain DNA, okay? So a virus is not going to be able to integrate RNA into our genetic material. Okay, so it has to convert the RNA into DNA so it can integrate it into our DNA. Okay, and then our cells will start to make viral proteins. It's not cytoplasm, no, it's kind of just like a space, the purple bit. The purple bit does not have a name. It's literally just like a layer of lipids, and then within that there's a layer of protein. It's a very simple structure, but it's definitely not cytoplasm. Yes, Kira, we're going to get into that now. Kira, you've obviously been to a lot of immunity lessons in year 12. Okay, we're just going to pause for a second then. We're going to do something easier, hopefully, before we get onto the hard stuff. Just lulling you into a false sense of security. This is another, well, it can be a pathogen, right? Viruses are pathogens, bacteria can be pathogens, they can cause disease. But a bacterium is a cell. It is a cell. Can we remember what type of cell? What type of cell is a bacterial cell? And then really quickly, just for revision, we're going to label it. It's not a eukaryotic, it's the other one. It's prokaryotic. Why is it prokaryotic and not eukaryotic? Good, Natalie, well done. Don't worry, our brains are getting a bit rusty over summer. Okay, it doesn't have a nucleus. It doesn't have a nucleus. Okay, its genetic material isn't in a nucleus. That's a really good way of saying it. Yeah, it also doesn't have other, like it doesn't have mitochondria. But It is still a cell, okay? Because it can respire. It can grow. I mean, we could list everything. It can reproduce. So it is classed as a living thing and it is classed as a cell. Right, as quickly as you can in the chat, because this is obviously a little bit aside from today's lesson. So as quickly as you can in the chat, just for revision, give me the letter and give me the name of this bacterial structure. So we've got A all the way through to I think. And I'm going to wait to see the correct answers, then I'm going to put them on. So A are not called cilia. Can you think of anything else? I'm going to write it next to the letter. I think I can just about fit it on. So B is plasmid, which is basically a loop of DNA. F is the flagellum, which is like a tail, right? It's used for movement. It's used for mobility. E, ribosomes. They look quite big, don't they, to be ribosomes? But they are meant to be ribosomes. Yeah, ribosomes for protein synthesis. D is cytoplasm. So you're starting to see it does have cellular structures. It has cytoplasm. It has ribosomes. And it has a cell membrane. So C is, yeah, the circular, or we could say loose DNA, because it's not in a nucleus, it's just loose in the DNA. Nabil, I wouldn't call it chromatin. Nabil, can you remember what chromatin is from last week's lessons? Can you remember what chromatin is? You could say genetic material, Aisha, but I don't think you would say chromatin. There's a reason why. Can anyone tell me what chromatin was? And we still need to get these layers in the right order. We've got 3 layers. Yeah, so chromatin, thanks Aaliyah. Chromatin is DNA wrapped around histone proteins. And I'm going to tell you now that DNA in a prokaryote does not have any histone proteins. That's one of the differences. So DNA in a prokaryotic cell is not packaged around proteins. You wouldn't be, I don't expect you to have known that, okay? So I'm not surprised you said chromatin. But chromatin is DNA plus protein packaged together. And actually, the DNA in a prokaryotic cell is not wrapped around histone protein, so we can't call it chromatin, okay? It's only eukaryotic cells that have those histone proteins. Guys, why are we avoiding the outer layers? Do we know which one is which? If I help you, so I is the cell surface membrane. So that's the inner layer. H, what would be next to the cell surface membrane? G is not the cell wall. It's something else that bacterial cells can have. Yeah, a lot of you are saying G is the cell wall. So H is actually the cell wall. Yeah. Good, Hamza. So G is called the capsule, or sometimes called the slime capsule. Okay, so you've got that third layer that can be around the outside of the cell wall. If we come back to A are called pili. Who's heard of them before? Or a single one would be called a pilus or a pilus. Who's heard of those? I think there was only one of you that actually put that into the chat. Might not have done them at GCSE. But yeah, not all bacterial cells have pili. They don't all have flagella. They don't all have capsules. But again, this is just like a generalized structure of a bacterium. Okay? Okay, so some of you haven't heard of them. Person one hasn't heard. So the pili are a little bit shorter and straighter than the flagellum, and they're used more for attachment. So the pili help the bacterial cell to attach and stick onto things, whereas the flagellum is used for movement. It propels the bacterial cell forward, okay? Now, this is obviously not a virus, but the point of doing this today was to show you, look, bacterial cells are cellular, they are living things. They've got lots of the same structures as a cell, like the cytoplasm, like the ribosomes, like the cell membrane, whereas a virus has none of those things. Okay, it's very simple. It's just genetic material, protein, and a lipid envelope, basically. Okay. Okay, so that was just a little bit of revision. Let's do something more difficult. Are we ready? Let's think about what HIV actually does. Reproductive system. We're not going to do the reproductive system. We don't do that anywhere in A-level biology, actually. Did you do it at GCSE though? You should have done it at GCSE, like labelling the reproductive system and the menstrual cycle. We don't do it at all on A-level, on OCR or AQA, interestingly. So, what is HIV? What does it actually do? We're going to get into now how it makes you ill, how it causes AIDS. Does anyone know what HIV stands for, by the way? I should have asked you that before. What is HIV? That's not good, Jawahir here says. No, it's quite an interesting topic that at GCSE, isn't it? So what does HIV stand for? It is sexually transmitted, but it stands for human immunodeficiency virus. Okay, that's where the abbreviation comes from, HIV. So you can tell by the name, it affects your immune system. Immunodeficient means your immune system is deficient. It's going to be weakened by this virus, okay? So, HIV infects a particular type of immune cell. Now, at A-level, we're going to call it the T helper cell. But for you guys who've only just done GCSE, I'm going to tell you that a T helper cell is a type of lymphocyte. What type of cell is a lymphocyte? Highly infectious virus. I mean, yeah, it is. So that kind of makes sense, HIV. Right, a lymphocyte is a white blood cell. Good. That's where we got to at GCSE. It's a white blood cell. WBC, yeah, when we're being lazy. So, but at A-level, and this is one of the topics I know you're going to find hardest in year 12, we do learn about different types of lymphocytes, okay? So there's one type of lymphocyte called the T helper cell. But today, let's just think of it as a lymphocyte, okay? So what basically happens is HIV infects these lymphocytes, these T helper cells, and basically hijack them. They turn them into a HIV replicating cell. They hijack the cell. It's like it becomes a HIV making factory, okay? So in a healthy person, someone without HIV that's not infected with the virus, there should be about 1 million T helper cells or 1 million of these lymphocytes in just 1 milliliter, tiny, tiny volume of blood. But basically what happens, and you can see this on the graph, take a little look at it now, if someone is infected with HIV, it will gradually reduce the number of these cells, so these T helper cells or lymphocytes. Now, if you imagine, if the number of lymphocytes in your body is going down, 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 you're weakening your immune system. And then what starts to happen, and you can see we've got years on the x-axis on this graph. So this individual, after about 9 years following infection, they started to get lots of early opportunistic infections. So opportunistic infections are just infections that by chance you pick up. So they might have more colds, they might contract the flu, they might contract pneumonia because they've got a weakened immune system, they can't fight it off. And then there's some later, possibly more serious opportunistic infections. And it's actually these infections that can be fatal. Okay? Now, not because, like in a healthy person could probably fight off the flu quite easily. They could fight off COVID quite easily. They could fight off measles. They could fight off pneumonia. But because the immune system has been so weakened and the number of T helper cells has gone so low, these individuals cannot fight off these infections. So they end up dying from things like pneumonia or more recently COVID because their immune system is so weak. Okay? Okay. So this is the hardest bit of the lesson. How does HIV replicate? I've given you a simple diagram, but I think we are going to add to it a little bit. I feel like you're up for it. We can add some more detail, right? So we can really learn this to A-level standard. So we've got our HIV virus. We know the structure of the HIV virus. We know it has RNA. And we also know it needs reverse transcriptase. Remember, that's an enzyme. Okay, so let's go through. Follow with me, please. Let's look at number 1. So HIV attaches to receptors on the white blood cells, on these lymphocytes. Can anyone remind me which part, this is where you need to answer my questions now, so which part of the HIV is it going to use to attach to the lymphocyte? Think about the structure of the HIV and what we said at the start of the lesson. Not pili, because it's bacterial cells that have pili. Remember, we've gone back to the virus here. Good. A few of you saying pili. I've confused you with the bacterial cell, haven't I? It's going to use the attachment proteins. Look at these green things. You see these here? These are the attachment proteins. Okay, so let's just add a few notes. It says HIV attaches to receptors. We're going to put using attachment proteins. Okay, so HIV can't infect any cell. It can only affect T helper cells, lymphocytes, because those cells have receptors. Can you see these things? This is the receptor. They have receptors on their cell membrane which are complementary to the attachment proteins on HIV. I'm just going to use my hand. So you've got a receptor on the surface of the lymphocyte. You've got the attachment protein on the surface of the HIV, and they fit together. Yeah, they're complementary. So the HIV attaches to the lymphocytes. Okay, let's go to number 2. So HIV then injects its genetic material. Remember, genetic material in HIV is RNA. So it's going to inject its RNA into the lymphocyte, which we're now going to call the host cell. Now, there's something missing from this diagram because it jumps straight to number 3. But what is missing? Once it injects that HIV, once it injects that RNA, what needs to happen to that RNA? Because we need to add that on for step 2. And this is where the reverse transcriptase is going to be used. So it injects the RNA into the host cell. Good. We're all following me. I'm so glad, because sometimes it's hard planning these lessons, right? Because I think, oh, is it going to be too hard? Are they going to be put off? Are they not going to want to do A-level biology? So I'm trying to pick stuff that's like interesting but not too hard, and then this is quite hard. So RNA is converted into DNA. And in an exam situation, you would have to name the enzyme. So using reverse transcriptase. Okay. Then we can also add on the DNA is now inserted into the host cell's DNA. I don't suppose any of you know the enzyme that does that, do you? Thanks, CFK. I'm glad you're joking. You don't even need to know the name of this enzyme in A-level biology, but there is another enzyme responsible for actually inserting the DNA that's made from the RNA into the host cell DNA. I'm wondering if Kira knows what it is. You don't even need to know the name of this at A-level. But I was wondering whether Kira had heard of it. No, it's called integrase. All enzymes seem to end in A's, right? Integrase or DNA integrase. It integrates the viral DNA into the host cell DNA. Okay, are we all following me so far? Are we good? Have we got any questions? So we've got the attachment protein on the HIV binds to the receptor on the lymphocyte, which is called a T helper cell. It injects its RNA into the lymphocyte, and it uses reverse transcriptase to convert the RNA into DNA. And then the DNA is inserted into the host cell DNA. So now you've got a body cell and it's got viral DNA in the host cell DNA. It's kind of slotted it in there, okay? Do we need to know how reverse transcriptase works? In year 13, you will do reverse transcriptase in more detail, but in this topic, which is a year 12 topic, no. You literally can just say reverse transcriptase converts RNA to DNA, okay? But you will learn a little bit more about it in year 13. Right, number 3. So we're up here now. HIV is then using the lymphocyte's cellular machinery to make copies of itself. That doesn't sound very scientific to me. So I'm going to just edit that slightly just to make it sound more sciencey. Basically what happens is the host cell will now make or manufacture viral proteins. And the viral proteins assemble to make new viruses. So you're basically hijacking the lymphocyte. You're getting the genetic material of the HIV into the host cell's DNA, and that cell is going to start making new viruses, okay? Can you please tell us more today? It's in a whole other topic though. It's in like genetic engineering is when you do it again, because we use this enzyme for our own purposes. We use it in genetic engineering. We use it to make human insulin, for example. I'm going to go off on a tangent now, but like, you know how we make human insulin for diabetics? We can use reverse transcriptase to do that because we take the RNA from a pancreas cell, we add reverse transcriptase to the RNA, and we make the DNA copy of the insulin gene, and then we insert it into bacteria so they can make human insulin. That's where you learn about it again in year 13, insulin. I don't know why, everyone always says I say insulin funny. Isn't that with plasma rings? Yeah, you can, once you've got the DNA for the human insulin gene, you insert it into the plasmid of a bacterial cell, yeah. But we actually use reverse transcriptase to make that insulin DNA. Okay, and finally, Now you've hijacked the cell. Now it's making HIV proteins and assembling HIV viruses. Once there's a certain number in there, it's going to burst. You're going to destroy the host cell. Okay, and then all those viruses, which you can see here, you can see them escaping. All these viruses are going to repeat the process. They're going to bind to more lymphocytes. They're going to inject their DNA or RNA and convert it into DNA. They're going to hijack another host cell. That host cell is going to be destroyed, and it just continues, right? After the initial infection, it just continues. So, because I'm very kind, and I do this quite a lot in my normal lessons, like if it's a hard topic, I give you like a model answer. So quite often on the A-level exam, they will say, describe how HIV replicates. This would be like a 5 marker. Does the DNA go through transcription translation? Yeah. So once the RNA has been converted into DNA, it's inserted into the host cell's DNA. And then obviously you've got the whole process of transcription translation to make the viral proteins. Exactly. So this is looking at mark schemes over the years. This question, how does HIV replicate, has come up about 4 or 5 times now. These are basically the key marking points. So you can see you don't have to learn everything. It's not too detailed, but they're focusing on the attachment proteins attached to receptors on the helper T cell or the lymphocyte. The RNA, I would say, enters the cell. Reverse transcriptase converts RNA to DNA. The DNA is incorporated into the helper T cell DNA. Viral proteins will then be produced. Viruses will be assembled, and they'll be released from the cell. Now, I've never seen this as a 6 marker. It's usually just 4 to 5 marks. Okay, so in reality, you would need to learn 4 to 5 of these statements. So they're making it a little bit easier for you to get good marks because you don't have to say everything. Okay, give me a little, how should we do it? Give me a score in the chat. Like if I gave you this question now, how many do you think you would remember of these bullet points? 1 bullet point, 2 bullet points? Would you remember 4 or 5? Give me a score in the chat. There are 6 altogether, but it's usually only a 4 or 5 marker. Jawahir says 3. 3 to 4. That's brilliant. 2 marks on a good day. I mean, I think the first 4 are quite easy to remember. That's the ones I would learn. 4 to 5, brilliant. So anyway, you've got that model answer now, so you can impress in year 12. When you're doing this topic, you can just throw out a perfect answer and get all of those marks. Okay, we are going to look at a couple of questions to finish. I don't think I've given you this question because I've literally just given you the answer, but I've given you some other questions, I think. So let's have a little go at these. Now, we're told in question 1 that this is HIV, but the bits that it's pointing to, A and B, all viruses have those bits anyway. Whether it's HIV or coronavirus or measles virus, they all have those bits, A and B. So put your answers into the chat for me. This is just a simple structure of a virus or HIV. And then I think I'm going to throw a C on there as well, because this is HIV, so I'm going to pick something specific. Should have waited for a few more answers then, shouldn't I? I've only got A and Zainab giving me the answers. Where's the rest of your answers? Good, so B is a capsid. Now, all viruses have these. So I've thrown on C because C is something that not all viruses have. They're only going to have that if they have RNA. So C is reverse transcriptase. Okay, because this is HIV. It has RNA as the genetic material. Okay, question 2. These are harder questions. Using the graph, explain why the number of HIV particles or virus particles in the blood rises during the first few months after infection. So I'm assuming infection happened here at year 0. This says time in years, by the way. So infection happened here. Now look at the green line. The green line is the number of HIV particles in the blood. We can see it's increasing. But why is it increasing? Why are there more and more and more HIV viruses in the blood? Okay, not a bad answer, Zainab. We're going to use the graph though. So Zainab, instead of saying they replicate inside host cells, can we use the graph? Look at the orange line on the graph. They've named a cell to help you and what is happening to the number of those cells. I'm going to use the first part of your answer though. Good. Aaliyah says the number of T helper cells is decreasing, which is telling us they are replicating inside T cells and destroying T cells. I can see this on the graph because the T cell number is decreasing. As the number of HIV viruses is going up, the number of T cells in the blood was going down. So it's kind of showing you these viruses must be using those T cells as host cells. They are replicating inside the T cells, they are destroying T cells, and then spreading in the blood. You could even say infecting more T cells. Okay, so yeah, and you can see, look how steeply the number of T cells is going down. So those of you that said HIV must be rapidly replicating are correct. They are destroying T cells at an alarming rate. So they must be bursting out of T cells, destroying the T cells, traveling in the bloodstream, infecting more T cells, destroying more T cells. Yeah, you get the idea. So we've got 2 marks. The idea that they're replicating inside T cells or destroying T cells. But once they do that, they're then spreading via the bloodstream or they're moving and infecting more T cells. Okay? Right, question 3. This person developed a large number of infections about 9 years after he first became infected with the human immunodeficiency virus. Using information from the graph, explain why. So let me just see if I can move my face so you can actually see the full graph. So 9 years. Is kind of here, right? So after 9 years, we are being told that this individual started to get loads of other infections. So common colds, flus, measles, bacterial infections, maybe COVID. They're starting to pick up a lot of infections, which could potentially be fatal for this person. Why is this? Has anyone got any suggestions? This is 3 marks. This is quite a hard question. I think one of these marks you probably won't be able to get yet, so I'll have to help you with it. But what has happened to the number of T cells in the blood? we can definitely get that mark. Look at year 9. What's happened to the number of or what is happening to the number of T cells in the blood? Good, Aaliyah. So at 9 years, the number of T cells is decreasing. Or you could say T cells are being destroyed. They actually allowed you to get a mark here, by the way, just for saying the immune system is weakened. But what you won't know, unless Kira does anyone know what T cells, helper T cells, does anyone know what they actually do in the immune response? Yeah, Aaliyah, I actually think you would have got 2 marks there because you've said the number of T cells is decreasing and therefore the immune system cannot fight against pathogens, which is the idea of a weakened immune system, right? Does anyone know what T cells do? This is in the immunity unit of year 12. So I'm not expecting you to know this, but, ooh, never done that before. I'm going to tell you. They do have something to do with activation. I'm probably gonna confuse you now right at the end of the lesson, but I'm gonna go for it. So T cells activate B cells. And B cells produce antibodies. They activate all the stuff, yeah. You're not wrong, Ella. Think of the name. These lymphocytes are called T helper cells. And the reason they're called T helper cells is they help to activate another type of lymphocyte, which is called a B cell. So T cells and B cells are both types of lymphocyte. They're both types of white blood cell, right? But T cells are needed to activate B cells. And B cells are the ones that make antibodies. So, linking this together, which is maybe the hardest thing we've done in this lesson, but if the number of T cells is decreasing, we could get a mark for saying there are fewer T cells to activate B cells. Okay, and then we can say fewer B cells means fewer antibodies. And you've heard of antibodies at GCSE. You know that antibodies are needed to help destroy pathogens, right? So if HIV is destroying our T helper cells, fewer T helper cells to activate the B cells, the B lymphocytes, fewer B cells to make antibodies, less antibodies means a weakened immune system. Okay? How do we feel about this topic? Guys, you've got a lot of this to come in year 12. I know from experience that the whole immune response, so the phagocytes, the lymphocytes, the B cells, the T cells, the memory cells, this is one of the topics that students find the hardest. But is anyone quite interested in that stuff? Is anyone looking forward to the immune system at A level? You love this topic already? Oh, great. I do. Because at GCSE, you learn it so basic. You literally just say, oh yeah, white blood cells, lymphocytes, phagocytes. Destroy pathogens. It's so basic. Now at A level, you learn exactly what a phagocyte does. You learn about different types of lymphocytes and exactly what they do. And you learn about memory cells and long-term immunity. And I think Jawahir said at the start that you like those kind of stories in biology. Well, the immune response is one of the biggest stories in biology because you go all the way from infection to the complete immune response, right? Destroying the pathogen. It's one of the longest A-level biology stories. We've just touched on it there, but hopefully it's piqued your interest and you're looking forward to it. Okay, Guys, it's pretty much time to finish. I hope you found today's lesson interesting. We've kind of strayed away from cells. We've done something a little bit different. So we've done HIV structure, viral structure, but we've also done how HIV replicates and a little bit at the end there about how it affects the immune response. Go and do your homework for me if you've got 10, 15 minutes. And I'm going to let you go, I think. I'm going to see you, when do I see you again? Monday? Thank you. Thanks, guys. I'm hoping you enjoyed that lesson. I've not put you off. It was interesting. Thanks, Kira. Thanks for all your great answers as well. Thank you. Right, I'll see you next time. Bye.